Welcome to Cramper University. It's great to have you with us. Thanks for joining us. My name is Toby Thompson. I'm the studio director in the glorious Grenville Turner Studios here on campus. We are here today to talk about biosurfactants for oil spill response. So if that's of interest to you, and we hope it is, then stick around. We're going to be here together for about an hour. If you're watching the recording, and hopefully you are doing that too, then welcome again. We want to carry on, EBIC want to carry on the conversation with you. I think we know that you know us, so let's find a way to carry on that conversation. Talking about questions, we want this to be an interactive session. We're going to be together for no more than 1 hour, so we want your challenges, your comments, your observations at pretty much any time. You know how Zoom works, you should know by now. So find the Zoom text chat and then type us a curious question, a challenging question, something around the topics that you're hearing from us today, just to get the juices flowing. Goes without saying, stay in touch with EBIC, and there'll be some introductions to upcoming events from EBIC later on at the very end, so stick around. Joining us in the studio, we're going to have 2 or 3 amazing experts in the field. I think you'll know some of them. You may even know their work. So we want to give you the opportunity to pose questions to them and find out really what's happening, what the latest in terms of biosurfactants for oil spill response is for you. We're going to kick off, and I'm going to introduce our first studio speaker in the presence of greatness, if I can say that. I hope you don't mind. Tony Gutierrez, it's great to have you with us.
It's been a pleasure.
Tell us about yourself. Kick us off by saying a little bit about your background, how you've ended up talking about this whole topic area.
Yeah, so I'm a professor at Heriot-Watt University in environmental microbiology and biotechnology. I've, for over 20 years, been working on oil degrading bacteria, the microorganisms that respond to oil and break this pollutant down into innocuous substances. I was in the United States when the Deepwater Horizon happened. And I did a lot of work on that spill since then. In fact, still doing work on the Deepwater Horizon spill.
The effects are still happening.
Well, the ecosystem has recovered in about a year's time since the spill occurred. It was very devastating because a lot of issues, problems, the economies that depend on the ecosystem and so forth. But my focus was on the microorganisms that responded to the spill. And I was there in the United States doing a Marie Curie fellowship on oil degrading bacteria. I arrived there towards the end of 2008, and the spill occurred in April of 2010. And I asked the European Commission that was funding me, could I stay a little longer? Because this huge opportunity has occurred. It's not often that you get such a huge spill like this, which was, in fact, it was unprecedented. In the past, we've had big spills, but nothing to this level, with the exception of the EBIC 1, which happened in the Gulf of Mexico, now dubbed the Gulf of America. But that's another topic. But it was unprecedented in the sense that, well, the volumes of the spill were huge, similar to the EBIC 1, but it happened at 1.5 kilometers under the sea surface. Something like this had not happened before. Deepest oil spillage in the history of the United States, but also the world. And the oil gushed into the Gulf of Mexico for almost 3 months.
Too long.
That was a very long time. And I got involved in the spill with the Institute of Marine Science at the University of North Carolina at Chapel Hill. Also got involved with other collaborators and so forth. And we've been doing a lot of work on that.
Oil in the blood. Don't go there.
You could say that.
Wow. If you're interested in what you just heard, queue up those questions. I'm going to come on to another speaker first. Let's introduce the panel before we start the conversation. Boyd, I'm going to come to you next if I can. How have you gotten into this whole field? Tell us about your job title, where you are, and a bit of your background.
Lecturer at Essex University. My background is quite unusual. I was working in finance when I left school in London in business banking. And the first oil spill that I remember was the Exxon Valdez in 1989.
Yeah.
And about 10 years after that, I decided to leave banking and went to Essex University.
Good for you.
To do a degree in marine biology. And I followed that up with a PhD that's similar to Tony, was in oil degrading microbes.
Because you thought it was a good idea.
I did, yeah, and it was something that I got into during my marine biology degree. I became very interested in molecular ecology and a lot of the tools that I still use today, working with DNA, RNA and proteins to understand how microbes work. And yeah, I've been working on it ever since, and that's since about 2004, so just over 20 years. It's been one of my main research areas, oil degrading bacteria.
And you do something which I barely understand, transcriptomics and proteomics. Go on, give us 10 seconds on that.
Yeah, so if we have a bacteria that can break down oil, it does it with enzymes, which are forms of protein. It holds the information on its genome, so we can use genomics to understand what a microbe can do. The transcript is when that's converted into RNA, which then gets translated into protein, which is actually the functional enzyme that much of the work I do is in those enzymes that are in pathways to break down hydrocarbons, which are the major components of crude oil.
So if you're into transcriptomics, we want to hear from you. Put something in the question in the text chat. Have one of us talk about it. Brilliant. Boyd, thank you. Stick around because we're going to come back to you.
Thank you.
One final speaker that I want to introduce. Rob, tell us a bit about your background and where you're from and how you've ended up into the field. You've been here, been OSRL for 23 years. That's a long time.
Yeah. So a bit like Boyd, I got a marine biology background from Bangor University in North Wales. And then I did a PhD down at Exeter University in conjunction with the Marine Biological Association and Plymouth Marine Laboratory, and that was specifically on very early studies in ecotox. And I was looking at the effects of oil spills and oil-derived hydrocarbons on rocky shore communities, so on those kind of shorelines. So that's where my sort of oil spill interest happened, and I got inspired by all sorts of incidents, including the Sea Empress and Amoco Cadiz, and like Tony, I was involved with the Deepwater Horizon spill. But yeah, so my particular interest is in the environmental impacts of oil spills and how we mitigate those through a number of tools in our response toolbox, which I can talk about. So yeah, that's my background.
And I love the fact that you talk about bridging science to response. Yeah. That's your passion.
Right? So spill response as a topic is science driven. You have physics there, you have chemistry involved, you have biology involved, you have microbiology involved, because ultimately oil is a pollutant when there's too much of it in a single place at any one time. But the ultimate fate of any sort of oil in the marine environment that we're talking about here today is the biodegradation pathway, which my esteemed colleagues here know a heck of a lot more than me at the sort of a microbiological level. I'm more on the sort of macro level. How do we mitigate impacts from a spill? Such
bit of judicious buck passing there, Rob. Rob, I'm going to hand the floor over to you. Kick us off.
Yes.
And it's just a reminder to the audience: if you have a query, a question, observation, or a challenge at any point in any of these people that we've just heard from, then put it into the Zoom text chat. Rob, over to you.
Yeah, okay, so yeah, here's my first slide. I've got about 4 or 5 slides that we'll go through. So I work for a company called OSRL, oilspillresponse.com. You can have a look at our website, loads of information on there, loads of background on who we are and what we do. But essentially, we're the world's largest industry-funded cooperative. So we're owned by over 150 oil and gas, energy companies. Some governments are also members of OSRL. So it's a cooperative where the members pay into a pot, if you like, which is our company. We're dotted all over the world, Middle East, Far East, America, here in the UK, where I'm based. And we're set up to respond to oil spills 24 hours a day, 7 days a week, with people, with expertise, with specialist equipment to do that. So yeah, as it says there on the slide, we provide resources to prepare for. So when we're not responding to a spill, we're helping our members and governments and lots of other stakeholders be prepared for an oil spill, writing plans, doing training, looking at doing environmental assessments. And then if there is an incident, we're here to assist as and when. Generally, we're set up for large international incidents where we cascade our resources into to bolster local and regional capability. So on the next slide, you'll see some of the tools in our response toolbox. So monitor and evaluate. We always have to know where the oil is, how much there is, where it's going to go. So we use a range of aerial assets to do that, people on the ground, on the beaches, surveying. We have computer models that can let us know where it's going to go. And then we can put in a number of different techniques that are driven by NEBA. So NEBA is a process. It means net environmental benefit analysis. And we use the tools in our response toolbox to help drive the best NEBA outcome for a spill. And the way we do that is we compare, if in an oil spill scenario, if we did nothing, this would happen. If we put in a number of mitigating techniques in place using some of the tools you can see here on this slide, then what would be the net environmental benefit? What would be the reduction in impact of that spill on environmental receptors, shorelines, habitats, socioeconomic, so fisheries, etc. So lots of people might be aware of things like mechanical recovery, which you can see top middle, which is basically booms, floating booms and skimmer systems to recover the oil off the surface. You can do in situ burning. That was done a lot during the Deepwater Horizon Macondo incidents. So that was a lot of in situ burnings taken place there. But one of the biggest tools in our toolbox we have is chemical dispersants, and they can be applied via an aerial asset, and we have a couple of these aircraft that you can see here, or they can be applied via vessels, or there is also one of the big things that came out of Deepwater Horizon was subsea dispersant injection. So injecting dispersant subsea at the wellhead that's having the uncontrolled flow event, and that's done for a number of reasons, and we don't have time to go into that today. But yeah, so in terms of today's talk is about biosurfactants, but we at OSRL don't use biosurfactants, but in terms of our bridging science to response activity, we're always curious about what's the next technique that we can bring to our toolbox in the future. And it may not be that biosurfactants could do this on the scale that we do, but there is oil spills that are happening all over the place all the time, and we're always looking at ways of bringing additional techniques to those particular spills. So on the next slide, this is some shots of our dispersant stockpiles. So we have these chemical dispersants dotted around the world, many thousands of cubic meters of these stockpiles, and there's many different types, but essentially a chemical dispersant does what it says on the tin. It disperses the oil. Now, oil will disperse naturally anyway during a spill into the upper water column. What chemical dispersants do is they accelerate that natural process that would take place, which then makes the oil into much smaller droplets, so they become more available for biodegradation. Like I said earlier, that's the ultimate fate of oil. It gets used as a carbon source, which Tony and Boyd will talk a little bit more about. But essentially, a dispersant has two elements. It has a solvent, which is the carrier, for a surfactant, and the surfactant has, you can see on this little image here, has a hydrophilic, so a water-loving part, and an oil-loving part, an oleophilic part. And when you add a dispersant to an oil spill, mix a bit of wave energy, it reduces the interfacial tension and produces lots of little droplets in the upper water column, roughly to sort of 50 to 100 microns diameter. And that's how we accelerate that natural dispersion process. So, and the way that we apply dispersants can vary as well. So the next slide shows the different application systems that we have, and others do have around the world. So you've got these vessel-based systems with boat spray, so like hose pipes, if you like, off the bow of a boat, and then you've got these spray arms, which are boom arms out of the side of the boat. But those systems are designed for the droplets to become a very specific size so that they're effective on the oil. There are some of these active boom systems as well that we use, but you can see there that we can have an encounter rate or an application rate of about 2 to 10 cubes per vessel per day. If you compare that to an aircraft, you can cover a heck of a lot more surface area with a helicopter or a Hercules. We have a Hercules out in Singapore, or we have 2 of these Boeing 727s here in Europe, and they can do many more sorties with a much higher volume of dispersant per day. And then, as I said earlier, we've got this subsea dispersant injection, SSDI and DDS, that was first trialed during Deepwater Horizon, and that's done at the subsea level. But it's really important that I just note that these chemical dispersants they're modern type dispersants. So the actual surfactants that are used in there is generally proprietary information, but they are no more toxic, if you like, than and are found in everyday items such as detergents and shampoos and that kind of thing. So if you imagine, breaking down oil in your dishwasher sink, they're using fairy liquid or any kind of detergent. That's how they use. So modern dispersants are very low toxicity, less toxic than the oil itself, but they're used in this NEBA process to accelerate natural dispersion at sea, generally offshore, deep water. And what we're trying to do there is reduce the risk of shoreline impact, because when oil impacts various shorelines, that's when you can get into trouble. So I'll pause there. The last slide is just a close-up of our Boeing 727. This is just in an exercise spraying water, which we do on a regular basis. So that's one of the differentiators for OSRL as a company, that we have this wide aerial dispersant delivery system out of the back of a 727, or a couple of those. So yeah, that's a quick summary of who we are, how we use chemical dispersants. I could talk for hours and hours about them, but we don't have that today. So yeah, so it's really about using our science to help mitigate environmental impacts of oil spills, but large or small spills.
Rob, that was amazing, and not least of which the resources that you can call upon. But I do have a kind of segue question for you before we go to Boyd, and that was, you're looking at dispersants, but what's the barrier to bringing biosurfactants into the response toolkit?
Probably one of the barriers is the volume that we need for very large spills. So how can you ramp up the production of biosurfactants? And I'm not saying that's never going to be possible, but at the moment we have these chemical dispersants, they're stable, we have them in large stockpiles. That's for big incidents. But biosurfactants may well have a role in much smaller incidents, because if you think about a very sensitive environment like a mangrove or a salt marsh, it doesn't take much oil to cause an environmental impact there. So a lot of people think that you need a heck of a lot of oil to produce a large environmental impact. No, small amounts of oil in a very sensitive environment can cause some very large impacts. So yeah, that's the context there for biosurfactants.
Brilliant, Rob. Thank you very much indeed. Stick around, because there's a question I can see already in the chat, which is coming your way. But we'll do that a bit later on if I can. Boyd, let's bring you up on the screen. I don't know whether you're able, Boyd, to respond to what we just heard there from Rob in terms of those barriers, but the floor is yours now. Over to you.
Yeah, I'd agree exactly with what Rob was saying in terms of scale for oil spill response. That would be the biggest challenge, because producing biosurfactants in that type of volume would be extremely tricky. But that's perhaps somewhere where engineering biology can come in to actually increase yields of biosurfactant that are produced by bacteria. That's something Tony will touch on a bit later. And with improvement in different types of fermentation strategies, hopefully biosurfactants can be produced at scales where they can be used for many different reasons actually, and not just oil spill response.
Kick us off, because my question in the back of my head is, why are they good? But really, Boyd, over to you.
Okay, thank you. So yeah, I just wanted to talk a bit about some of the work that me and my research team do at Essex that's all related to oil spills. So we do a lot of experimental work in the lab, but as Tony mentioned in the beginning, we also work out in oil spills as well. In fact, I mean, even though major oil spills have reduced significantly over the years, there are still many spills of varying sizes that happen in the marine environment. And almost every PhD student I've ever had, during the 3 years of their PhD, there has been an oil spill that has ended up becoming part of their thesis, because like Rob responds to clean it up, we can also respond to go and sample it. And we've actually worked with Rob on oil spills and oil spill science for many years at Essex. So some of the pictures you can see there on the top left, that's actually just proving that I do go out in the field. That's me in the yellow hat.
I wondered who that was.
Yeah, I'm actually in the middle of an oil slick in the northern North Sea there, which was actually one of the only experimental oil slicks to actually test dispersants at sea. So I've seen firsthand dispersants working, and the effectiveness of them is quite amazing. You know, it instantly disperses oil into the water column, gets it off the sea surface, and allows microbial bacteria in particular in the marine environment to biodegrade that. In the center, that kind of satellite image, the black you can see on there, is an oil spill on the Athens Riviera that was from a tanker going down. And it's if down on the bottom right, that's a little Mediterranean bay, and depending on the color, that looks like nice sort of low energy seawater, but that's actually thick crude oil that has washed up in a bay in a very sort of low nutrient environment. So, you know, oil spill response was involved in cleaning that up, but we were also involved in getting an urgency grant from NERC to actually track the biodegradation of that spill over many months after. The one down on the bottom left is the Thames estuary, and I put that picture on there because that's kind of where I did my PhD those many years ago. It used to be the site of some big oil refineries. There was a lot of oil traffic, fine sediments in the Thames estuary, very interesting microbial communities associated with oil biodegradation. It's actually where the Port of London is now based. So a lot of those refineries have since shut. And on the top right, that meandering creek system that you can see is entering Poole Harbour, and that was from a spill a few years ago that actually in the UK, it was a pipeline spill under the creek from an oil producing facility. And Rob's company were involved in the cleanup, and again, we monitored this site for many months to do post-spill monitoring, looking at the microbial communities and I'll show you a little bit of that in some of the coming slides. So if you pop onto the next slide, I'm just going to show a few of the things that we kind of do at Essex, and this one is actually from that spill. Excuse me, my
don't worry,
earphone has just fallen out. And what you can see on the top left here is this is called an ordination plot. It looks quite a complicated figure. It's to actually reduce microbial data into something easy to read. So we sequenced millions and millions of pieces of DNA from the sediment environments where this oil spill was, and taxonomically assigned information to them to see what types of bacteria were in different environments. Essentially, every single dot that you can see on that plot is a complex microbial community. And simply, if the dots are near to each other, it means that that microbial community and the genes that within it are similar. And if the further they're away, it's how dissimilar those microbial communities are. So without going into too much detail, all the ones that you can see in the middle of various colours are areas around that Poole Harbour where there was no oil or very small amounts of oil that was rapidly cleaned up. But on the bottom left of that top plot, where you see the pinkish purple ones, these are where the oil spill actually happened. So what that's telling you is the microbial community are very, very different in that site. And if you just scale down to the graphs that are showing 2-ring, 3-ring, 4-ring PAHs, for example, those are polyaromatic hydrocarbons. So we use a chemical technique to measure those in the environment. You can see the structures of some of them on the left. These are toxic and carcinogenic hydrocarbons. So obviously there's great interest to remove them from sediments and seawater. And all those structures can be degraded microbially by bacteria. And we can use the information that we do from DNA sequencing to actually, I mentioned identifying some of the bacteria. The ones that you can see on the little small square plots on the right are just a few examples. If anyone's interested, they can read the paper, the details of it are underneath. But this is showing the ones in purple at the oil spill site are largely elevated in that region. So we can start to see which type of bacteria are growing. If you look in the paper, we can also see which bacteria don't like oil and are affected and reduce in abundance compared to all the other sites that were not oiled. But we go well beyond that and actually start to look at the enzymes as well in that sequencing. So the paper will show that in this environment, it's a low oxygen environment. Most of these bacteria are anaerobic, and that means without oxygen in anoxic sediments, and they need very different pathways for hydrocarbon degradation. And the image that you can see at the very top that says premium, this is the post-spill monitoring, so post-incident guidelines that are created by CFAS, and we have a small piece in that on how microbes can actually tell you something in post-spill monitoring. So this is evaluating the sites many months after to see when they're remediated and also tell you something about how the oil hydrocarbons can actually be biodegraded. And if you pop onto the next slide, please. And as Rob mentioned, dispersants, we've done a lot of research on dispersants at Essex as well. Here on the top left is just a very simple diagram of how Rob very nicely explained that they work. So the application of a dispersant, either from ship or a plane, will disperse that oil into the water column. And it seems like a physical process, but actually, you know, this is a form of bioremediation, because if that decision is made to use a dispersant, we're actually making the decision to let bacteria deal with the oil and naturally attenuate it in the marine environment. And we get a huge increase in surface area to volume of the oil as it's dispersed into droplet phase. It's very rapidly colonized, each droplet, by bacteria to biodegrade that oil. So what you can see on the top right is a couple of commercial dispersants. So we've done some experiments with those. And really the idea of biosurfactants would be to potentially replace dispersants with something more natural that has a bit of a greener footprint. But mainly because also they are easy to biodegrade, because many bacteria, simpler structures, and there's enzymes that already exist that can break down many of the biosurfactants that bacteria produce. So if a dispersant wasn't sprayed, the hydrocarbon degrading bacteria in the sea would actually produce their own biosurfactants to do the exact job that that dispersant is doing, but it takes a bit of time. And just to say that, you know, the chemical surfactant market is huge. They're used in all sorts of products. So biosurfactants are becoming in demand, particularly for many other industries such as drug delivery and personal care items and, you know, green washing up liquids, for example, where surfactants are used. Next slide, please. And this is some work one of my PhD students did on dispersants themselves and biosurfactants. The plot that you can see on the top left, Rob mentioned they break interfacial tension between oil and water. This is a simple measurement of the surface tension of seawater, which you can see on the left at around 70 millinewtons per meter. And the effect of chemical dispersants, which have great names such as super dispersant and slick-on, reduce that massively to round about 30, and that is what drives the reduction of that surface tension, and interfacial tensor will drive the oil into the water column. And what this is showing is we've got a range of biosurfactants produced by different bacteria, rhamnolipid that Tony will talk about in a bit, sophorolipid and trihalolipid, and many of them are just as effective as chemical dispersants in driving the oil into the water column and creating a stable oil in water emulsion. And the plot to the right of it, this is the abundance of bacteria in oil contaminated water with those different surfactants and chemical dispersants in. And the one on the left where it says oil only, that contains no dispersant or biosurfactant, and the rest are the first three are commercial, following that are biosurfactants, and then the next three commercial dispersants. And this is showing the amount of bacteria, so really the amount of growth that we see, and it's at a 24-hour time point after oil has been added. So it's a log scale, so we've got sort of 10 or more times growth of bacteria in a 24-hour period. And the red fraction of the bar is actually OHCB, stands for obligate hydrocarbonoclastic bacteria. And these are bacteria that we know can degrade many different types of hydrocarbons. There's lots of different types, some alkanes, some aromatics. And you can see that a much, much larger proportion, almost 100-fold in some cases, within 24 hours of bacteria that can actually degrade those pollutants. So really efficient. And we're doing some work in Essex now on EBIC. You can see the team there. So Hannah and Monica are currently doing some work to isolate a new novel hydrocarbon degrading bacteria, of which many have been done so far. And we can have a simple screening process to see the effectiveness of biosurfactant production, which we can measure in the lab, and then pick bacteria to actually work with and go on to perhaps try and identify genes and pathways that are involved in biosurfactant production for later downstream processing and engineering biology. The next slide, please. And this kind of just summarizes some of the ways we work. So I mentioned at the beginning proteomics and transcriptomics that we can use. That on the left, the shotgun analysis that you can see is a method of extracting proteins from an oil spill, fragmenting them and then searching them against known genome databases to identify what enzymes associated with oil degradation we can find. And we use a whole host of different bioinformatic packages to then predict function of different enzymes and use different databases. And that gives us the opportunity to then work with those bacteria to create modified organisms, so engineering biology, and that can be deleting genes or putting genes in to improve function, or use the bacteria for the creation of bioproducts. So one of course, these biosurfactants, but there's also other interests in oil degrading bacteria because some of them can produce what can be used as bioplastics as well, such as polyhydroxyalkanoates that are produced as storage compounds. And also we can use the genetic machinery to make biosensors as well. And the two plots that you can just see on the right, some work done by Ben, one of my other PhD students. This was identifying a pathway for biodegradation in a cold water marine bacteria called Oleospira. The cloud of dots that you can see on the top right, these are proteins. The further up it is the more significantly different it is. The further to the right is how much it was overexpressed relative to a control, which is the same bacteria growing on something that's not a hydrocarbon. And that feeds down into the plot below where we can start to piece together enzymes in the different pathways. So these are alkanes, which are linear hydrocarbons. They're quite inert, not very chemically or biologically reactive. So the first step, an enzyme will introduce oxygen. That's what sets these bacteria apart from most other bacteria, that will convert it to a primary alcohol, and other enzymes take that further through to an aldehyde and a fatty acid before it enters through kind of central metabolism that many bacteria have. And the byproducts are carbon and energy source for the bacteria, and CO2 and water are from an alkane, the only outgoing products. And yeah, on that note, I think that's the end of my slides.
And on that bombshell, there is a question, Boyd, for you, and this is from Zulfikar. There is a question for you from Zulfikar, and this is around indigeneity. And I'll read it out, so pin your ears back, get that earpiece back in your ear there, Boyd. Given that many indigenous hydrocarbon degrading bacteria can naturally produce biosurfactants, Zulfikar asks, do you think future oil spill response strategies should focus more on stimulating native biosurfactant producing microbial communities, biostimulation for instance, rather than applying externally produced biosurfactants? And what are the key ecological or regulatory barriers to implementing such a process in real spill scenarios?
Yes, it's a really good question. One of the issues that we obviously see when oil comes into the sea initially is that it does take a while for bacterial communities to respond. They do start to grow quickly if there's no other resources limiting, but there is always a lag phase. And part of the reason is that in clean environments where hydrocarbons are minimum, they are usually at low numbers. So a surfactant can actually help increase that growth in the very early stage. And if you think of a slick at sea, one of the main reasons for dispersing it is to stop that oil actually hitting a coastline where we know it causes a lot of damage. And the dispersants do stimulate those indigenous bacteria, but they save them the energy of producing the biosurfactants themselves, so they can start to grow very rapidly as the oil's dispersed. We often see a lag phase of 2 to 3 days for many bacteria to produce the biosurfactant themselves and then grow to the numbers that can actually degrade it that quickly. Adding a biosurfactant shouldn't really pose too much regulatory hurdle. Adding an engineered microbe certainly would, but this is why initially we're interested in producing a product that could actually be, put into the sea to kind of help that. And the biosurfactants themselves, one of the ones Tony will talk about, many of them come from marine bacteria that produce these in situ. And in some of our experiments, we have also seen self-recognition. So if you add a biosurfactant, you can actually encourage the growth of the particular type of bacteria that produce those surfactants in nature. So there's possibility in the future to have biosurfactant formulations that contain quite a few different types that can stimulate the types of organisms that you want to in different environments.
Boyd, thank you very much indeed. I've heard Tony's name mentioned several times already, as if everyone's going to come back to you. I don't know if you have, before you start in your sessions, you have any idea around this whole indigeneity thing, if you have an opinion about it.
Yeah, we can come back to it. Yeah, through the slide presentation that I have. Sure.
Brilliant. We're going to have some slides for you. So when you're ready, go ahead.
Yeah, so the first slide that you'll see here is of the Deepwater Horizon spill.
You'll recognize that.
Which caught the world's attention. It was in the news globally. It almost equally like the Exxon Valdez.
2011, have I got that right? 2011?
2010, April 2010, yeah. And it stopped the research of a lot of research groups in America, as well as around the world, to focus on this spill, because it was just an enormous opportunity to work on this, understand what's going on geochemically, what's happening to the oil, the microbial response, which is something that I got involved with. And yeah, so maybe if we just pop to the next slide, just showing you here the volume of what an oil slick looks like that's on the sea surface.
It looks like sand dunes, but that's in the sea.
You could look at it that way, yeah. But this is, so I wanted to show this slide because this is one of the purpose of using chemical dispersants, is because one of the main concerns with the oil reaching the surface. Remember, it was the oil gushing from down 1.5 kilometers on the seabed, gushing into the water column, and it reached the sea surface, of course. Some of it formed into a massive cloud at about 300 meters depth. It formed this huge cloud that was traveling in a particular trajectory, southwesterly. That was also tracked and studied in terms of the microbial response in this cloud.
And the cloud was never going to surface?
No, it was dissolved oil in the subsurface. Microbes responded to that. It caused an oxygen anomaly. So oxygen was depleted, of course, because the microbes were using the oxygen to hydroxylate the hydrocarbons to break them down. A lot of papers came out, a lot of studies came out to do with that in tracking the plume. Eventually, they lost track of the plume, so we couldn't do much else to that with studying that. But of course, the oil also reached the sea surface. The concern there was that these huge amount of volumes of oil could reach the coastlines, sensitive receptors that Rob mentioned, salt marshes and so forth. The oil did impact the coastline and impacted beaches. That caused its own devastation to bird life there and so forth. But the purpose of using a dispersant, and it was Corexit, a generic name, Corexit, that was used. There's other different Corexit types and so forth, but I'll just use the word Corexit.
Some of the stockpile that we saw from Rob's
chemical
images.
dispersant, exactly,
Right.
yeah. So that was applied on the sea surface.
Aerially?
I believe it was aerially.
Right.
Rob could comment on that later. But it was used to break up the oil and dissolve it back into the water column to stop it reaching the coastlines. But some of it, of course, eventually did. The Corexit was also applied down at the wellhead where the oil was gushing out.
How was it applied? Submarine?
Incredible engineering. Yeah, incredible engineering to get down there and apply the dispersant. And that was also to dissolve it within the water column before it reached the sea surface, which still some of it did, as you can see by these oil slicks here. And the purpose of the dispersant, as Rob mentioned, is to dissolve the oil to enhance the activities of oil-degrading bacteria. That's the sort of knock-on effect after the main reason being to prevent it from going to the coastlines. Can we have a look at the next slide? So we mentioned about biosurfactants. I've been doing research on biosurfactants for more than 20 years now. And marine bacteria, like terrestrial bacteria, some of them produce biosurfactants. Some are more powerful than others, which means they're more effective at dissolving oil, breaking it up and so forth, which is great for the bacteria to break it down. One of the ones that we study and that we produce in the lab is rhamnolipid. This is a type of biosurfactant. It's considered a very powerful biosurfactant. It has commercial applications, not just for potentially as an application to oil spills. It hasn't been used in oil spillage, of course, because Rob mentioned about one of the major bottlenecks is producing at a large scale. So that's a major bottleneck in terms of getting biosurfactants to be applied for combating oil spills, is getting the bulk. You have to grow these bacteria up in fermenters. Fermenters, well, they can be expensive, but a lot of the costs are the ingredients that go into fermenters, maintain the fermenters, particularly the downstream processing steps. Removing after a fermentation has taken place, removing the cells, taking the spent medium, which is the supernatant, the liquid medium, which contains the surfactants, the biosurfactants, and then the downstream steps of purifying those surfactants, that costs a lot of money.
So that can't be industrialized, or it needs another investor?
It can, but it's very expensive.
Right.
So if something's expensive, who's going to take that up? It's not economically viable. In niche commercial applications, like potentially cosmetics, drug delivery, that sort of thing, where it can be economically viable because the final end product you can charge a premium amount, then yeah, that biosurfactants are applied there. But for things like oil spills, not. We did a study. So this is a paper here on your slide that it was done by my previous PhD student, Kristina Nikolova, who lo and behold is now working with OSRL.
No surprise there.
And she did incredible work. I mean, she was absolutely brilliant PhD student who published a lot out of her PhD. Not only book chapters, but reviews and original results studies. But this paper in particular, published in the journal Microbiome, she compared rhamnolipid with a chemical dispersant called phenosol. in its to understand the effects on the microbial response. So she went up on research cruises into the Faroe Shetland Channel, which is a region up north of Scotland. So you've got the Faroe Islands and you have Shetland and the Shetland Isles, and in between you have a channel. And in that channel you have a lot of oil rig extractions. So there's a lot of, the oil industry is omnipresent there. So what are after the work I did in the Gulf of Mexico, Gulf of America, if you will, I came here to the UK, take up my faculty position, and I wanted to study more about oil response, bacteria involved in biodegradation, production of biosurfactants, and all that sort of thing. And what a perfect study point, the Faroe Shetland Channel. And so Christina did this study where she compared rhamnolipid with finosol. The results were quite stark and really, really interesting in the sense that the rhamnolipid produced a microbial response and it enhanced the activities of oil-degrading bacteria, including these obligate specialist oil-degrading bacteria, which, why are they called obligate specialists? It's because if you give them your typical sugars like glucose and the sucrose and these sort of things that you would expect bacteria to absolutely love, like fish and chips to us, they don't touch them.
Weird.
They won't even touch them. They don't have the genetic capacity to do that. But if you give them oil, that is like our fish and chips. They will thrive on oil.
Was that happenstance that you found that out, or
No, this has been known for a while. It dates back to to work of Boyd and the older academics, Ken Temis, Peter Goloshin, Misha Yakimov, all these people that studied oil-degrading bacteria and discovered a lot of these oil specialists.
Wow.
My work has revealed some other additional oil specialists as well. That was work that I did at the Scottish Association for Marine Science and subsequently UNC Chapel Hill. But from her results, Christina, going back to Christina's results, she showed that rhamnolipid stimulated particular specialist bacteria for which were suppressed by finosol. And the results were evidenced further when we looked at the degradation of the hydrocarbons and showed that the rhamnolipid favored the degradation of aromatic hydrocarbons, which are more toxic than the alkanes, long-chain hydrocarbons, whereas the finosol did not induce the degradation of the aromatics. So that was actually quite a stark, surprising result, which, you know, it's to say biosurfactants do have a future, but it's about, you have to, the problem is that bottleneck, which with getting large scale compared to the chemical dispersants.
I want someone to talk about that, how you get that technology readiness level up to producing at vast scales more of that. I don't know whose role that will be other than someone with a lot of money. Wow.
Can we go to the next slide?
Yeah.
So this slide here is a
looks like a constellation in the sky.
It is, isn't it? It is, and we have described it when I've entered this image into competitions. I've described it as some sort of galaxy or whatnot. But this shows you what dispersants can do and what biosurfactants can do. This was actually a biosurfactant produced by a marine organism. It's not a rhamnolipid. It's more of a polyglycolipid, or I think it might have been a glycoprotein actually. But it's produced by Halomonas that I isolated during the Deepwater Horizon spill. And it just shows you that, so we simulated an oil spill in the laboratory, applied this biosurfactant, and you can see here it broke up the oil into teeny tiny droplets. The scale of this, some of these droplets are 1 to 2 microns, and even less than 1 micrometer.
The scale just looking at that.
Right. So some of these droplets are less than the size of a bacterial cell.
Very small.
And this blue, cloudy, white coloration is the formation of what's called marine snow, but in this case, marine oil snow. So it's exopolymeric substances, which are dissolved naturally in seawater, coming together and forming this marine snow.
You didn't expect to see that?
Yes, I did, because in the marine, so during the Deepwater Horizon spill, this was another unprecedented event where marine oil snow was formed in huge amounts
given the volume
on the sea surface, and it eventually sedimented down to the sea floor.
Wow.
So these mucilaginous, fluffy materials that were formed during the spill, And what was interesting, so we did a further study on this, and we showed that in these agglomerations, oil degradation occurred at a higher rate than in the surrounding seawater environment.
What you want.
So that's that. So what inspired us to do further work on this, and from these results, is that potentially in the future, when there's an oil spill, would it be worth to apply some sort of, whether it be a biosurfactant or some sort of chemical dispersant that can induce the formation of marine oil snow, which are potentially a hotspot for increasing the speed of the degradation of hydrocarbons in the water column. So that was another interesting thing from the work that we did.
Christina,
here.
I hope you're watching this, or I hope you're getting news of this, you being bigging you up. I guess you don't want there to be another oil spill, that you can have an opportunity to explore that, but I guess it's incumbent upon all of us involved today to do that.
Yeah, so one thing I said, it was an incredible opportunity. That sounds fantastic, but we don't want to see another major spill like that, because they are horrifically disastrous. Yeah. Can we? Yeah. Next. So this next slide.
It looks like my lava lamp at home.
This next slide shows you pretty much taking a marine oil snow agglomeration, like in the previous slide, putting it under the microscope under epifluorescence microscopy, and labeling the bacterial cells in that agglomeration with a fluorescent probe.
Wow.
And so these tiny little dots that you see glowing are the bacterial cells within the agglomeration, breaking down the oil at a faster rate than in the surrounding seawater. Those large green globs are the oil, so they fluoresce naturally as well. But the tiny dots, the important point here, these are the bacteria breaking down the oil at a faster rate than in the surrounding seawater. So, in the future, potentially, we might be looking at, there could be an approach here to, instead of just applying a dispersant or biosurfactant to break up the oil and speed up the degradation of the oil in the water column, maybe to induce the formation of marine oil snow, these agglomerations, where they act as a hotspot to speed up degradation.
Wow. I've got to come to Rob, because it seems to have come full circle. Rob, do you want it to snow? It sounds like you could use, Rob, some of these things that we've just heard Tony talk about.
Yeah, absolutely. Marine oil snow definitely caught our attention with some of the outputs from the science from Deepwater Horizon, and to that end, we worked with Boyd on a PhD for it. So that's all again about that bridging science to response. We wanted to know more about this marine oil snow phenomenon. But yeah, absolutely. The all this science that my colleagues here have spoken about could potentially feed into the response techniques of the future. And that's why, as an oil spill scientist in a response world, we remain very open and flexible and never closed to new ideas for the future.
You need those new ideas. Stick around.
Yeah.
Rob, there's a question, I think.
absolutely.
Bidgie has a question for you. It's quite a long one, so I'm going to read it out if I can. And there's a kind of question in there, so I'll let you pick out the question. Thanks for this. I completed my PhD. This is Bidgie. Completed my PhD at Sultan Qaboos University in Oman, where my research focused on heavy crude, upgrading using biosurfactants and oil spill bioremediation. Although the work resulted in high-yield biosurfactant production at both laboratory and pilot scale, including successful spray-dry-based large-scale processing, companies were reluctant to adopt biological surfactants in the field, preferring conventional chemicals. I think you know the story. And steam-based upgrading. I'd still love to know more about biosurfactants reaching a large-scale industrial application. Rob, I guess that's been the recurring theme here. How do you scale this up?
Absolutely, you need chemical industry involved. You need the oil and gas industry involved. We need spills of opportunity where we can prove that technology with experimental opportunities in spills. So yeah, we're also talking to a university here in the UK about how we can use enzymes produced by bacterial communities to help with shoreline response and breaking down weathered oils and some of these new low sulfur fuel oils. So we as an industry remain curious on how we can do that. But it is a challenge to break the norm, I suppose, of what we're used to and the techniques that are tried and tested. I guess it's a bit, the analogy is a bit like electric vehicles, that we're encouraged to use, but not everybody, not all of us want to take them up. We're quite happy with the vehicles that we have, and we don't want to get into EVs. It's that kind of analogy. So my job with numerous colleagues within our industry members is to open eyes, and we do that at OSRL conferences and other events, and we work closely with Boyd on those kind of events. We get the PhD students to come and talk about their research in our world, not in the academic world. So we try and that sort of cross contamination, if you like, of from one industry to another. And it's great to hear Tony talk highly of Christina, who's now a valued member of our team here. So yeah, thanks, Tony.
Let's hear for Christina one more time. Brilliant, thank you. I just love, Rob, I love your enthusiasm. Boyd, can I come to you, because your name's been mentioned several times. Can you talk, we talked about challenges and barriers earlier on. What have been your main challenges in engineering biosurfactant pathways?
Well, at the moment, the work we're doing at Essex is more in discovery phase to look for new ones. One thing that became really apparent to us is there are very few bacteria in the marine environment where we actually know the genetic pathways for the production. The rhamnolipid is probably the most well studied, and the Pseudomonas organisms that produce that's been known for some time. One of the main obligate hydrocarbon degraders, alcanovorax, alkane eater in the sea, the genes responsible for producing the biosurfactant were only discovered last year. So the databases are quite small. That Poole Harbour spill that I mentioned, we interrogated all the sequences, and this is millions of DNA sequences from the environment, against the known surfactant databases, but there's only a few bacteria that are in those, such as Pseudomonas, Bacillus, Rhodococcus, but there are many, many more. Every oil degrader that we work with, we know produces a surfactant, but we still have the challenge to actually work out what are the genes responsible to be able to manipulate those pathways. But I think the challenges going forward really are scale, and it comes back to what Rob was saying in terms of testing. It's easy to show in the laboratory experiments, such as Tony and myself have talked about, how effective they can be. The next stage would be to sort of step that up a bit, and if you can start to produce at higher yields, it wouldn't take much to then put that in the only outdoor testing facility for dispersants, which is in New Jersey in America, and this is a large wave pool where you can test dispersants. That's to look at the efficiency of it actually dispersing oil into the water column at a larger scale. You can't study the bacteria in that because the seawater is chlorinated, and then obviously eventually testing in the field, which can be a challenge, because as soon as you actually disperse oil into the water column, it can be quite difficult to follow that and monitor the development of bacterial communities, because it disperses rapidly to sub-part per million concentrations, and you put oil from the surface into a 3D environment that then moves with depth and width with the energy that's in the sea. So it transfers the oil over a very, very large area, which has the benefit of it being biodegraded, because by reducing the concentration of oil that contains the hydrocarbons, the nutrients that the bacteria need, such as nitrogen and phosphorus, it can take those up at a much lower carbon concentration so that growth isn't limited.
Gosh, Boyd, we could talk about salinity tolerance at some point, but we have run out of time. Hopefully you've got an appetite for more of this. Thank you for taking time to explore. I wanted to explore some more, but we can't. We know that we have some events coming up in the EBIC calendar, so go to the EBIC website and you'll see that there's another one of these webinars events coming up in June, so please log on to that. You'll see the QR code up on the screen now. And there's also the conference later on this year, Early Career Researchers Conference. We have several early career researchers in the audience here today. It's been fantastic having your questions and comments. Engineering Biology for Environmental Solutions, and we're accepting abstracts now. So get your abstract in. So please scan that code. In the seconds that we have remaining, it just allows me to thank Rob. Thank you very much indeed for joining us all the way from Southampton. Give us a wave.
Pleasure.
I've really enjoyed your passion there. Boyd, likewise, we could keep on talking for a lot longer. And Tony, goodness me, there's some amazing pictures. You should be an artist. I think you are an artist deep down. But if you like anything that you've seen here, please get in touch with all of the speakers in the usual channels. Thank you for watching. The producer today was Nicola Heaven. The studio manager was Chris Ripley. My name is Toby Thompson. Thanks a lot.